Welcome to solverfy.
Hello students, the question asked is describe how the proton gradient is used to make ATP. The proton gradient is created during the light-dependent reactions of photosynthesis when protons are pumped into the thylakoid lumen, and this creates a high concentration of proton inside the thylakoid and a low concentration outside. And these protons then flow back across the thylakoid membrane through ATP synthase, which uses energy from this proton flow to synthesize ATP from ADP and inorganic phosphate, which is ADP plus PI becomes ATP through the ATP synthase with the help of the proton flow. Another question asked is, what two molecules bring chemical energy from the light reactions to the next stage of photosynthesis, which is the Calvin cycle? The two molecules that bring chemical energy from the light reactions to the next stage of photosynthesis, which is the Calvin cycle, are ATP and NADPH, which are synthesized during the light reaction and are used during the dark reaction, which is the Calvin cycle. In the third question, we have been given a diagram which shows that actually it is the Calvin cycle which shows different stages or phases, phase 1, phase 2 and phase 3, and we have to name these phases of the Calvin cycle. The phase 1 here represents the carbon fixation, in which carbon is fixed, and the second phase represents the reduction phase, while the third phase represents the regeneration of carbon dioxide acceptor, which is the ribulose bisphosphate. In the first phase of the Calvin cycle, which is the carbon fixation, carbon dioxide here is incorporated into the organic molecule through a process called carbon fixation. The enzyme ribisco catalyzes this reaction between carbon dioxide and the 5-carbon molecule, which is the ribulose 1,5 bisphosphate. producing a 2,3 carbon molecules of 3-phosphoglyceric acid. While increase of phase 2, which is reduction, the 3-phosphoglyceric acid molecules are reduced to glyceraldehyde 3-phosphate using energy from ATP and electrons from NADPH. And some of these glyceraldehyde 3-phosphate molecules are used to make glucose. And other organic molecules, while continue to the next phase, which is the regeneration phase. So in case of regeneration phase, the remaining glyceraldehyde 3-phosphate molecules are used to regenerate the ribulose bisphosphate, which can then accepts carbon dioxide and continue the cycle. Another question asked is, what enzyme catalyzes the reaction in the carbon fixation phase? The enzyme that catalyzes the carbon fixation phase is the ribisco, as already stated that ribisco, which is ribulose 1,5-bisphosphate carboxylase or oxygenase. I hope you found the video useful. Thank you.